Hello and welcome to this webinar titled Current USP Perspectives on a Rapid Sterility Test, hosted by Biopharma Asia magazine and presented by Dr. David Roesti, Technical Steward Microbiology, Manufacturing Science and Technology at Novartis, and Erin Patton, Senior Product Specialist at Charles River. My name is Orsolya Balog and I will be the moderator. Before we begin, I would just like to remind our viewers that there will be a live Q&A session at the end of the presentation. Audience members can send their questions at any time during the presentation via the Questions tab located at the right-hand side of your screen, and we will go through these at the end of the webinar. Now, please allow me to introduce Dr. David Roesti, Technical Steward Microbiology, Manufacturing Science and Technology at Novartis, our first speaker. Dr. David Roesti holds a PhD in microbial ecology from the University of Neuchâtel, Switzerland, and has 20 years of experience in the field of microbiology within various domains: drug product manufacturing, food microbiology, biogas production, microbial interactions in the rhizosphere. Currently, he works at Novartis Pharma AG in Stein, Switzerland, in the Manufacturing Science and Technology Department, and is responsible to define the microbial control strategy at the site, and is a global subject matter expert in microbiology for the Novartis Group. Prior to this assignment, he led the Rapid Microbiological Methods team at Novartis Pharma AG and was the laboratory supervisor for the microbiological testing of non-sterile drug products at Novartis Pharma Stein AG. Dr. Roesti is an elected member of the General Chapters Microbiology Expert Committee of the United States Pharmacopeia 2015-2020 revision cycle and is a member of the advisory board of the European Compliance Academy Microbiology Group. Finally, Dr. Roesti is the main author or co-author of many different publications in either peer-reviewed journals or book chapters, and has regularly held presentations in scientific congresses or expert groups. Welcome, David.
Thank you, Ursula, for this very kind introduction. I would also like to thank Biopharma Asia for giving me the opportunity to present the actual proceedings of the United States Pharmacopeia Microbiology Expert Committee on the Rapid Sterility Test chapter. Probably most of you in the audience are aware that when you have to perform release testing of a parenteral application, for instance, you apply the compendial sterility test based on the USP 71 methodology. Now, this method is quite an old method since it was initiated in 1936 in the USP, and it's composed of a growth-based method of two different growth media. One is TSV, the other one is FTM. And after having taken a sample tube product, filtered it, and introduced a nutrient media, you incubate for at least 14 days. And there are some products which are naturally turbid, and in this case, you might need to do an additional subculturing, which makes it even more longer than 14 days. So these methods, where it is totally acceptable for the conventional sterile drug products, which have a shelf life of 5 years in some cases, it is really becoming an increasing issue for the new advanced theoretical medicine products. Because now there are more and more new therapies which are coming on the market. It's great for the patients because these are really breakthrough therapies that really have a very powerful healing effect on the people. However, they are much more shorter shelf life. So there are now many cell-based products that cannot be cryopreserved, whereas they might lose potency. And in this case, the shelf life can be sometimes less than 2 days or 24 hours. Now, there are some other products which are widely used in hospitals in medical imaging, like positron emission tomography. preparations, these PT preparations. And since they have radionuclides, which have a shelf life of sometimes of less than 2 hours, and then the product itself has also a very short shelf life, they would also not really be adequate for a compendial sterility test, because sterility is 14 days. Obviously, you provided the product to the patient before the sterility test result is there. And there's an additional point that can be made for these special preparations is the batch volume is extremely small, so it's really less than, it can be less than 1 liter. So the sample volume which is required in the sterility test chapter is not adequate because it requires too much of a sample volume. So the advantages of having a compendial rapid sterility test would be that, first of all, microbiological safety needs for these short shelf life advanced therapies and PT products would be met with this release test. Also, if this is really an official rapid sterility test, the user does not need to perform a complicated and lengthy cross-validation comparing the equivalence of the rapid method with the actual compendial method based on USP 1223, for instance. The rapid sterility test may also be implemented without prior regulatory approval of the test method if this is indeed endorsed by health authorities such as FDA. And also for the instrument suppliers, if they know a test method is adequate as a rapid sterility test, they can base their instruments on this methodology, and then for them to penetrate the market will be much more faster because people will be using this method on a routine basis. So in order to address this rapid sterility test chapter, the third thing that the Microbiology Expert Committee did is that they set up working expert groups. So there was a modern microbiological methods expert panel which were raised, and the idea is to have really experts from different backgrounds and different stakeholder groups. So we had experts from the sterile component pharmacies, from the positron emission tomography labs, from cell therapy units, and also in the traditional pharmaceutical manufacturing areas. And here's the list of the experts. We also had some people which were representing the FDA on the team. So there was a lot of discussions within this expert panel, and basically the goal of the expert panel was to define user requirement specifications for rapid sterility test, and also to already have a pre-selection of potential candidate technologies that may be used to fulfill these user requirement specifications. The outcome of the discussions have been written in a stimulus article, and this is based on the modern microbiological method expert panel report. So you can still read this stimulus article on the website of the USP. I put the link here. And this stimulus article actually serves as the basics for a new USP General Information Chapter, the USP 1071. So the idea of the USP is first to write a General Information Chapter on the rapid sterility test in the first place, and in the second step, it would be to define a compendial sterility test that could be, for instance, used as an addendum of the USP 71. So the USP 1071 will contain different sections. It will contain a section on these user requirement specifications, on the concepts of risk-based microbiological monitoring and release testing, also situations where the actual compendial chapter is unsuitable for product release testing. It will also provide example of some technologies which may be suitable for these kinds of tests, a small paragraph on method suitability testing, and also a final paragraph on the compatibility of the technology selection with the different product categories. If we take a deeper look in the user requirement specification, so here I'm going to show you the most important ones. So they were categorized based on priority, and these are the ones that I'm going to show you now. First of all, obviously, the first one would be as short as possible time to result, and preferably prior to administration of the product to the patient. Then the second major user requirement specification is the limit of detection, which should be preferably less than 100 CFU. The system should have the ability to detect a wide range of viral microorganisms. The sample quantity, so the system can also test small sample quantities, so it does not consume a too large amount of the product. The idea is also to have this rapid sterility test methodology in different labs, and there are some labs we don't necessarily have an isolator to perform the test, so it would be preferred to use a closed system in this case to avoid cross-contamination from the testing environment. Then in order not to have a non-proprietary system and all people are really bound to one supplier, is to have the availability of instruments and reagents from multiple vendors. The availability of reference standards will be there because people will need to make some calibration or some qualification work on their equipment in a GMP lab. Then the system should be ease of use, should be easy to use, should be simple, so you don't need really a sophisticated analyst, which has a many years of training, and should be a very clear-cut result, and should not leave too much room for interpretation. We're working in a GMP environment, so it should really have a very clear-cut result. The system should also have a low false positive negative rate, and it should be possible to perform the suitability testing for each specific product. Also, the system should be really well advanced in the development, so it should be robust and there should be reliable equipment and reagents used in the testing. This is also from an engineering perspective. The system should not be a kind of university-based system, but really a system that has been produced by expert engineering teams. If possible, also sample preparation should be suitable for both manual and automated methods. Now, if you move and take a deeper look to the main user requirement specifications, the first one, time to result. So what do we mean with fast time to result? So it depends actually on stakeholder groups. So based on our expert discussions, there were actually different time to results, and that depended on the stakeholder group. So the products which required the fastest time to results were the PT products. Some radioisotopes has a very short shelf life of less than 2 hours. So in this case, time to result of 12 hours, less than 12 hours would be optimal. For cell therapy facilities and cell component facilities, generally time to result of less than 48 hours was considered acceptable. And for the conventional pharmaceutical manufacturers, less than 7 days. So obviously, for the conventional pharmaceutical manufacturers, it's not necessarily a question of patient safety. In most cases, it's really a business need, because if you can release your product fast on the market, you reduce also inventory costs, and then you have a certain business advantage. Now, there are some other advantages for patients, especially in the vaccines production. So if you have a pandemic and you can shorten time to release on the market of a vaccine, that really becomes also interesting from a patient safety point of view. For the specificity, so the idea here is that the system should detect a wide range of microorganisms, so bacteria, yeast, and molds, not just these typical compendial test strains which are listed in the Pharmacopoeia chapter, but most importantly, the microorganisms which are found in infection outbreaks or product recalls or in different sterility test failures of these products in question. So they should really focus on the bugs that can be found in the manufacturing process and also in the products of these stakeholder groups. Then the sample size was also widely discussed, and here the idea actually is to align with what is written in the Pharm Europe 2627 chapter on the microscope examination of cell-based preparations. So this chapter mentions 1% total volume of preparation to be tested for a product which has a low batch size. So here the idea would be to align on this and also to require at least 1% of the total batch volume. Now the limit of detection was really the URS which spurred the most discussions within the team. So there were two really different opinions. So some of the experts were the opinion that the limit of detection of this rapid sterility test should be non-inferior to the actual compendial test. It should be at least one colony-forming unit. Now the concern with this is that it might really limit the choice to some growth-based methods, which may require more than 48 hours incubation time. On the other hand, some experts were the opinion that the limit of detection should be validated totally independently of the compendial test, and higher than one CFU of detection may be tolerated for some real-time tests. So the concern here is if you have a limit of detection which is higher than one CFU, that might result in non-detection of low-level contaminants that might infect the patient. Now, however, several points to consider for the limit of detection. Now, the compendial sterility test has its own limitation. It can only detect a few microorganisms which are culturable under the test conditions of the sterility test. I mean, many people know, if you've been making comparison studies, that FTM is not the best media to recover anaerobic microorganisms, for instance. The sample size is limited and statistically weak when we are talking about really large batch sizes of more than 20,000 units, for instance. And on the other hand, for low volume cell therapy products, the sample size is also not adequate because it requires too much sample volume. Also, there's not really evidence that the actual limit of detection of the compendial test is really one CFU. If you make an analysis and you want to take a mean number of microorganisms of one CFU in a certain volume, generally microorganisms are distributed according to Poisson distribution in a volume of sample. So that means if you want to detect always one microorganism in more than 95% of the case, you need at least 3 microorganisms per volume that you are testing. There's also some evidence that the infection rate is not necessarily one CFU. So there's been a study, many studies which have been performed, and amongst them a study of Jacob and co-workers who really screened different platelet concentrates that were given to human cancer patients. And based on the metadata analysis, they suggest that infectious dose may be actually 100 to 1,000 viable microorganisms and not one microorganisms. Okay, so based on these different user requirement specification, different technologies were selected. So as I mentioned before, they had to be non-proprietary. Here on the table I'm showing, we can see a comparison of different methods which can be used. Limit of detection is really optimal case. It's when you have a very clear matrix with the best microorganisms possible. So generally, this is very optimistic numbers. Generally, the number is higher than that, but it just gives an idea of the comparison of the method. So in the methods that were selected, we selected growth-based and non-growth-based methods. We can see the detection can range either from 1 to 10 microorganisms, 10 to 100 microorganisms, and also depending on the culturability of the method or not, the incubation or the testing time can last from a few hours until 7 days. Obviously, direct detection methods would be the fastest methods, and some methods can actually both provide very fast detection and also a sufficient LOD. The USP 1071 chapter will also provide some clarification on the risk-based concept, because when people applying rapid sterility tests, they should also make a risk-benefit analysis, because there are some cases where the use of a rapid sterility test is more beneficial to the patient than staying with the actual companion test. So there's not a perfect method for the moment which is at the market, not one fit for all. You don't really have a method where you have very high sensitivity and almost immediate results. That's almost impossible. In just very rare cases you can apply this, but in most cases you cannot do this. So you always have, let's say, a certain weakness in the model that you're going to use, but sometimes that might not be relevant in what you want to demonstrate. So as an example, for instance, depending on the purpose of the application, you may have really clear benefits of rapid sterility tests. Now, there are some cases where there is a substantial risk to the patient survival when the product is applied too late. So there are really last results therapy. Every day counts in this case. So the faster you can provide the product to the patient, the better it is for him. And there has been some literature reports that when there are blood infections, every daily delay in providing antibiotics increase the mortality rate by 10%. So this is really, so every hour counts in this case. So if you can really provide the answer that your product, okay, it's clean, it's sterile, prior administration of the patient, and provide as possible for the patient, there is a clear safety benefit for the patient here. There are some growth-based methods which can perform continuous reading. So there are COT, for instance, detection methods which can do this. So instead of having to wait for final reading or perform intermediate readings every day, when the detection sensitivity is reached, microbial growth will be flagged by the system. And then an alarm can be immediately raised, so the people can really then react as fast as possible. So you can imagine, for instance, you have a product which is tested, and fast-growing microorganisms might be detected quite early on in the process, so within 24 or 48 hours. So if then the product can be determined, okay, it's not sterile, this would also ensure that the clinician can intervene sooner with the patient. Also, if you have a growth-based method, you can then directly cultivate your microorganisms again, or perform direct identification of the microorganisms to really focus on the best antibiotic treatment you can provide. There are also some rapid sterility tests which don't require incubation. So then you really have a direct detection. In most cases, the that detection will be above 1 CFU in this case. But there might be some cases where it's better to perform the pre-screening to verify that there's no gross contamination. So at least there will be an idea if there is a gross contamination or not before the product is applied to the patient. There are also some cases where antibiotics are directly given in the product formulation to prevent microbial growth, but there are some microorganisms which can survive with antibiotic formulations, and they might not be also detected by cultural methods. And there are also culturable, viable, not culturable microorganisms which would not be detected with growth-based methods. So there, these direct detection methods might be more powerful in this case. Now, one is thinking of just using rapid methods as a release test, but it really makes a lot of sense to use rapid methods during the manufacturing process as an in-process control, for instance. Because there, in this case, the faster you are with the detection, the faster you are with, okay, you can make an investigation much faster. And in some cases, you can restart production even faster. So that means also, in terms of the availability of the product to the patient, will be accelerated in this case, in case there's an issue with the product. Now, the best way, of course, and the fastest way to release a product to the patient would be to use parametric release. So not to perform a sterility test, but base yourself on the sterilization parameters, like for instance in heat terminally sterilized products. The idea in the USP 1071 is also to have a suitability test which should be performed for each product to be tested. So it's more or less based on what already exists in the actual compendial sterility test. So to spike the product with less than 100 CFU of a different set of microorganisms, and the test method should be able to detect these microorganisms which have been spiked. And however, on the other hand, if people are using direct detection methods or non-growth based methods or methods based on fluorescent labeling, for instance, there might be some background, inherent background from the product, and this has to be verified. So it really has to be verified there's no inherent background of the product to ensure that microbial growth remains detectable. And finally, there will be some explanatory on this General Chapter 1071 on the compatibility of the technology selection with the product, because there are some technologies which exist which work extremely well with some products which don't work at all with some other product types. So obviously the first one that people think of is the filterability of the product. Some products are not filterable, so all rapid methods which are based on filterability of the product cannot be used. Also, some products which produce naturally some fluorescent compounds. There are some cases where fluorescent based methods cannot be used. Also, some products like cell therapies may produce already ATP, and ATP based rapid methods may not be used in this case, or you need to have some kind of sample treatment. So I compiled here a few examples from the scientific literature on the compatibility of different technologies with different product categories. So in this table, you have direct detection methods. Interestingly, the qPCR can detect a wide range of, can detect microorganisms in a wide range of products as compared to flow cytometry, for instance. Then on the growth-based methods, so there you have different kinds of detection systems. And interestingly also, here is that the respiration-based system, so CO2 detection system, can also detect the whole spectrum of products like the conventional, filterable, non-filterable products, the cell therapy, blood plasmas, and also even some radiopharmaceuticals in some cases. So ATP bioluminescence, I will not talk in this in detail, so Erin will present a system based on ATP bioluminescence after my talk. So the next steps for this rapid sterility test. So the first step is to finalize the draft of the USP 1071 General Information Chapter and propose that this draft is then available for review in the September-October pharmacopoeia form edition. Then the second step, and this is going to take much more time, is to make proof of concept studies with different collaborating laboratories. And the aim of this is really to generate data that will help the USP define test parameters for a companion rapid sterility test. So in this second step, there might be some cases where some of the selected technologies that I showed you before might not be applicable at all for a rapid sterility test, but at least data will be generated to really evaluate this thoroughly. So I would like to thank my colleagues which are working intensively on this rapid sterility test chapter. So David Song, Tony Kundell, Ed Tidswell, and Hadati Al-Malai. I would also like to thank you for your attention during this presentation, and I'm going to give my word back to Ursula.
Thank you so much, David. Now again, I would like to remind our viewers that there will be a live questions and answers session at the end of the presentation, and the audience member can send in their questions at any time during the presentation via the questions tab located at the right-hand side of the screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second speaker, Erin Patton, Senior Product Specialist at Charles River. Erin received her Bachelor of Science degree in Biology from Xavier University and her Master of Science degree in Cell and Molecular Biology from the University of Illinois, Urbana-Champaign. Erin has over 14 years' experience in molecular biology research, microbiology, QC, and regulatory compliance. As a Senior Product Specialist for Charles River's Microbial Solutions Division, Erin provides innovative rapid micro solutions for endotoxin testing, microbial identification, and microbial detection. She is based in St. Louis, Missouri. Welcome, Erin.
So I wanted to talk a little bit about some of the topics that Dr. Roesti has already touched on, and thank you for a very nice presentation. It's always nice to hear about the USP Expert Committee activities. As such, I wanted to visit some of these topics myself and to tie that into some of my own experience with Charles River. So I wanted to come back to the stimulus article put out there by the expert panel, the Modern Microbiological Methods Expert Panel that was mentioned just earlier in the presentation, and tie into the piece here which states that one of the critical user requirement specifications that's been noted by the expert panel is that of the detection of a low number of microorganisms, that is the limit of detection. Of course, this is a very important consideration as we think about what might qualify as a compendial rapid sterility test. So let's take a little bit of a look at it. As presented back in the fall of 2017 by Dr. Roesti himself at PharmaLab at that conference, this particular user requirement, the limit of detection, was discussed quite a bit, and he just mentioned this earlier. There are a couple different sides to to the discussion here. One, that was stated that the limit of detection, or the LOD, of this rapid sterility test should be demonstrated not to be inferior to the compendial test as it stands right now. So we should see equivalency with that test. And as such, it would be expected with a rapid sterility test that the limit of detection should be 1 CFU. Of course, there is a concern that having a CFU that's down to 1 and having the sensitivity of the assay be that low, this may require some incubation time for the assay, thereby making the rapid sterility test just a little bit less rapid. You know, the opposing side to this to consider would be that if a higher than 1 CFU limit of detection would be tolerated for this test, then maybe the sensitivity is not quite so low, but maybe the overall benefit to the patient would be higher. And so we at Charles River would agree with this. We would agree with both sides of this discussion because both raise excellent points. Of course, if we're calling this a sterility test, it would be expected just traditionally that limit of detection should be 1 CFU. That would be critical. However, if a more real-time answer is required, like the example given with, particular compounded preparations that have short shelf lives, or something like a radiopharmaceutical, which has a quite short shelf life, then, maybe a real-time answer is the greater priority, and the limit of detection of 1 CFU is not quite as much a priority, in which case a higher limit of detection could be permitted. I would maybe consider this something like a screen. Of course, maybe it doesn't meet the definition of a traditional sterility test, but that doesn't mean that it would be without value to do such a test. Of course, because there's some kind of compromise here, and overall patient welfare needs to be considered, an approach like this with maybe a screen would need to be supported by a well thought out and well explained risk and benefit evaluation. So I wanted to touch a little bit on Celsis today. Celsis is a technology that's often referred to in the literature as an enhanced or amplified ATP bioluminescence. And so when I use Celsis, I'm talking about the Charles River platform particularly. I wanted to show that Celsis can actually be used in both ways. So respective to both sides of the discussion here, with a rapid sterility test with that low CFU of, or the low LOD of just 1 CFU, or just a screening test, Celsis is compatible with both of those types of approaches. So it has been shown that with regard to equivalence to the compendial sterility test, Celsis can perform with that level of sensitivity, and it has been shown that Celsis can achieve a limit of detection of just 1 CFU. It's been shown in several instances. We, of course, are aware of lots and lots of those, but some of them we have seen presented at different global conferences, and so I wanted to share an example of that with everybody. We have a few here. The first from PharmaLab in 2016. This is Novartis. The second one here in the middle, this is also from PharmaLab at 2016, and this is from a Janssen presentation. And then the last here, the one we see in the front, was from the ECA Microbiology Conference in 2017, and this is the laboratory called Labor LNS. And you can see here, it's very small, but the interpretation of results in each of these 3 conclusion slides shows that when Celsis was utilized for sterility testing, rapid sterility testing, that results were demonstrated to be equivalent with the traditional or the compendial sterility test. And so, also to go back to the other side of the discussion here, and think about, is it worthwhile to implement a test that maybe does not have quite as sensitive an LOD and can get down to 1 CFU, but maybe a real-time result is the more important thing, and that LOD of 1 is not as important. Of course, again, back to thinking through a risk-benefit evaluation or justification, there are situations like the examples that were mentioned by Dr. Roesti earlier in his presentation. Maybe that faster result is more imperative, and so this is doable with Celsus as well. Some modifications to protocol would need to be made, but of course, a very quick test would be able to be carried out. And so we would also recommend that maybe in lieu of only doing such a screening test right at the point of release or right prior to release of the product, that maybe you could round out this type of approach, this screening approach, with in-process controls. This would provide more comprehensive information about all those different microbial controls that are put in place when a drug is compounded or manufactured, that would give increased assurance that a finished product wouldn't have failed that traditional testing, that typical sterility test itself. So taking all that into account, it's great that Celsus has been designed with a very high level of flexibility. The method itself, we'll see in just a little bit, is very flexible, but in addition to that, the range of applications which may be served by Celsus is really quite diverse. So you can see here that sterility, which we're focusing on quite a bit today, that Celsus can be used for sterility, and not only that final product sterility test, which is so often thought about, but the maybe in-process control tests, sterility tests. So maybe this would be tests of bulk material prior to packaging. This could be even testing of incoming raw ingredients, any of those in-process tests that might be performed just to round out your picture of your quality assurance or your sterility assurance for your final product. In addition to being able to perform a sterility test, Celsus can also be used to perform a microbial limits test or a bioburden test. And again, it's not just final product microbial limits testing that may be done with Celsus. It's also the in-process control checks, just like I mentioned with the in-process sterility test. In-process bioburden may also be performed with the Celsus system. On top of that, the Celsus system and the way that it works is such that it's not a destructive test. It is a growth-based assay, and as that's the case, the enrichment that's used for the testing is reserved, such that if a retest needed to be performed or some kind of investigation, this is always a possibility because the enrichment persists. So let's go back to the stimulus article put forth by the Modern Microbiological Methods expert panel and look again at these user requirements. One of the other user requirements that was put forth to be looked at was sample preparation. And of course, this is an important user requirement specification because not all samples may be prepared in the exact same way. Different samples have different needs in terms of, maybe they could be filtered or not filtered. And Dr. Roesti had mentioned that maybe because of this, there will not be a single be-all, end-all rapid sterility test that's identified, but there will be considerations that need to be taken as individuals or companies select the methodology that will be right for them. And so, again, I think it's great that Celsus has been designed with a high level of flexibility, and not only in the different types of tests that may be performed, so the sterility test, the microlimits or the bioburden tests, screening kind of tests, but also the protocol itself is very, very flexible. So when we're thinking about the test method, if we start down here in the bottom left corner, we can work with samples with the Celsus method that require direct inoculation or membrane filtration. Maybe there's a requirement to filter away some preservative that could be in the sample, and if that is the best way to prepare the sample, that's suitable to do with the Celsus technology. Direct inoculation is also possible, so maybe you are thinking about an instance with a medical device, in which case you can't filter that medical device, but you would be able to put it into a container with broth to be able to perform an enrichment, a growth step. Both of those are perfectly capable of working with Celsis. Further, the sample size really does not matter so much with Celsis. We can the Celsis system can work with quite small or quite large volumes of sample, or even, like I say, not even necessarily liquid product, but different formats of product, even ranging to a medical device itself. The culture media that are capable of working with Celsis are very variable as well. So with sterility, we're thinking about usually the traditional media types of FTM and TSB, but if we're thinking of testing more like a microbial limits or bioburden test, other media types could be used, like lithene, TAT, lots of other options that are out there. And then the incubation time, again, based on what type of assay we're doing, sterility, maybe a sterility screen, in which case that real-time result is very important, or microlimits test. We could look at different incubation time to be able to validate that the test provides the results that are required within a particular time frame. And so just to give a few examples of some of the different kinds of products, because I always think it's fun to see what kind of products that are out there that have been tested with rapid systems. Again, we see that we can do product types that require filtration or maybe cannot, maybe are not compatible with filtration, different broths, things that are soluble or not, different pHs, products that are oil or water-based, pigmented, preserved. These are all compatible, at least with the Celsis system. And so to give more specific examples, things that would be more respective of pharma would be IV solutions, antibiotics, vaccines, even things that are, tablets, suspensions, lotions, ointments, some of these medical devices. The more interesting ones, although not so specific to pharma, I think it really goes to show just how many different industry types are interested in rapid sterility testing and how important it is that we continue to pursue rapid sterility testing as an industry, and also to show I think the flexibility of the method. It's so neat for me to see that samples such as toothpaste, printer ink, pudding, juices, things like that are capable of being tested, at least with the Celsis system. And so let's go back one more time then to the stimulus article that is recommending different technologies to be evaluated. Of course, the article has given some of the different criteria that should be evaluated, and those are important, as we've already talked about today. One of the final things that's mentioned right here in this abstract is the list of the different technologies, and Dr. Roesti had mentioned some of those too. And you can see here that I've done a little highlight to show that ATP bioluminescence, among others, will be evaluated as one of the candidate technologies for a compendial rapid sterility test. And so just to give a quick overview of this, there are different platforms out there that utilize ATP bioluminescence, and this is something that maybe if we think back to traditional ATP bioluminescence, maybe everybody can remember from school days that this is a mechanism, or this is an enzymatic reaction that has been understood for quite some time. This is not something that any of the ATP platform manufacturers has invented. Traditional ATP bioluminescence is based on the premise that ATP, or adenosine triphosphate, is present in all living cells, and that includes bacteria, yeast, and fungi. The standard ATP assays are using the enzyme luciferase to catalyze that microbial ATP into light production. And then any light which is produced is measured using a luminometer. Celsis in particular is a little bit different, and I mentioned before that often you'll see that it's referred to as enhanced ATP bioluminescence, or even amplified ATP bioluminescence, and you'll see here that I've called it by the reagent name, or the proprietary name of AmpiScreen, and this refers to the reagents in particular with the Celsis system. This is an interesting improvement that's been made on the traditional ATP assay, in which case we have a proprietary enzymatic reaction, in which case we have microbial enzymes that are converting a DP, so the diphosphate form, into ATP. And because of that, we see amplification of ATP to levels that are much higher than those which naturally occur. And that allows for a greater signal from each and every cell that was present when the test was performed. And because of that, we see the ability of the Celsis assay and the AmpiScreen reagents to achieve a very low limit of detection, as was presented before, where we can get down to that limit of detection of just 1 CFU. So it's a nice advancement in terms of sensitivity of the assay. The reagents themselves are, of course, they don't stand alone. The reagents work together to form the Celsis system, and so along with the AmpiScreen reagent kits, we do have the 2 different luminometers. That's the Advanced 2, which is seen here at the top, a little benchtop model, and the Excel, which is, I would say, almost a tiny benchtop model. Both are the same systems. They're both luminometers. They just differ in the amount of samples that may be tested at one time, so just a throughput difference. That works in combination with the proprietary Celsis software. And so it's the system combined with the software, the system, the machine itself, the reagents, and then of course the protocol, which is developed for whichever type of assay one would want to perform. Sterility test, maybe more like a sterility screen, microlimits, bioburden, whatever the need should be. And so, you know, we're so happy to have a system like this that could be evaluated by the expert panel, and a technology like this that's very flexible. You know, we understand, as was mentioned by the expert panel or by Dr. Roesti just earlier, that there are a lot of considerations and even limitations to keep in mind with the different systems. It was so nice to see that the table laid out just to outline, you know, strengths of different systems. And of course, for any companies that are thinking about going this direction, and for the USP panel too, it is so important to keep in mind considerations of the particular products that need to be tested, you know, the product attributes themselves and what kind of limitations those might present to any particular sterility system. And then, of course, consideration of the lab in which the testing is going to happen. How experienced are the personnel? This can play into whether one might want to adopt a more streamlined or more complicated technique or technology. The lab space, this can be a real concern when you think about footprint and throughput needs. You know, high throughput testing or maybe a lower throughput test. These are all important practical considerations, the logistics here, but they're important ones to think through as you decide on particular systems for specific applications. And this all rolls into considerations about the different technologies and vendors. So, you know, I just want to conclude by saying that, you know, we're really happy to see this movement with the USP expert panel outlining the different considerations that need to be kept in mind, beginning to look at selecting different technologies for evaluation, and hopefully that work will get underway before too long so that we keep moving the industry in a very positive direction towards rapid sterility, because really that focus with the microbiological testing should be on improved patient safety and improved product quality. If we can do that faster to make things you know, more beneficial to the patients, especially with those tougher situations, in which case answers are needed very quickly, you know, all the better for you know, for the health of those folks. So that's the last little bit that I have for everybody here, and of course we will be happy to address questions. I'll hand it back to Ursula.
Thank you so much, Erin. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that you can still send your questions via the questions tab on the right-hand side of your screen. Our first question is for David. Will the actual USP 7.1 chapter be replaced with the new content on rapid sterility testing?
So no, because the basic reason is that the actual companion chapter is harmonized chapter between the European Pharmacopoeia, the United States Pharmacopoeia, and Japan Pharmacopoeia. So we cannot just, add too much different information in these chapters. The idea would have it to have either as a complete separate chapter or to use it as an addendum, like the USP 71.1.
Thank you so much, David. The next question is for Erin. What type of controls do you run with the Celsius assay?
Sure. So controls are performed with each and every run, just to give the highest assurance that the test is performing as it would be expected to. And so negative controls are run just to ensure that there's not any kind of interference, within maybe some media or things like that, giving false signals. So this can give you assurance that you're not looking at a false positive result. There are also positive controls that are run where an ATP is measured, just to be sure that the reagents are performing as they would be expected and detecting any ATP that is giving a signal. So we have controls both directions to make sure that we can be confident with any results given.
Thank you, Erin. The next question is for David again. From the USP's perspective, is there an actual method of choice for a rapid sterility test?
No, so what we have done, we have listed the methods that could be potentially used as a rapid sterility test method. It will really depend on the purpose. So, and this the USP cannot decide because there's so much different product categories that we cannot, have really one method which fits every case. And also we would also get much more information in the next step of the project, where we will set up some collaborative laboratory studies and test then different types of product categories and different rapid methods. So we will also probably focus a bit more, limit the number of rapid sterility test systems, but for the moment there is no preferred choice.
Thank you, David. The next question is for Erin again. We have a product that we filtered during the sterility test, and it shows turbidity in the initial container. So we need to re-inoculate it, which elongates the incubation time to 21 days. Could we avoid this additional step by using Celsius?
Sure. So, these tests in which case there needs to be a secondary transfer of that first broth enrichment into a second broth enrichment, just to assess whether one is reading true sample microbial turbidity, or maybe just some sort of turbidity from the product itself. These can be very difficult for an analyst to read visually, so being able to use a rapid sterility test, or even just any kind of more advanced technologies to do sterility tests in this way can help. Not only does it cut the time, because turbidity is not the main mode of reading this test, it's really the ATP instead, which is a signal given by the cell, whereas turbidity can be caused by all sorts of things. And so, yes, it will cut down on that secondary transfer, such that you would only be looking at a one-time sterility test, not having to do the secondary test and extending out the number of days. So it can cut even more days off of the test situation like that, making it very, very rapid, I guess.
Thank you, Erin. The next question is for David again. Could other technologies as the ones described in the stimulus article be evaluated?
Yes, so the technologies are evolving. So this was the actual status 2016, 2017. Developers, system suppliers are introducing new technology. Some are more or less advanced, some are really at a very early phase of development. So The USP remains open in case there's a new technology or system which has an interesting aspect, and we can really use it as a rapid sterility test, and it's not included, we might include this also in the end.
Thank you, David. Our next question is from the audience. Would rapid sterility testing apply to sterile compounding in which the beyond use date is just a few hours, or this kind of high-risk sterile compounding is exempt from sterility testing?
So we had experts from the compounding centers on the team, and they've mentioned that they would accept to have a time to result of less than 48 hours. There are high-risk products where you have to apply within 24 hours already. If it's within 24 hours, there are a few systems which exist where you can detect your contamination within 2 to 3 hours. However, if you're really mentioning an almost immediate application, let's say within 2 hours, it doesn't really make sense to make testing for release prior to patient application, because by the time you deliver the product to the QC lab and the time you get the response, it's really too late. So either you can use rapid sterility test if you want to have a sterility test result as fast as possible, even if it's after the application of the patient, because in case there is a contamination, then you can provide antibiotics sooner the patient. You can perhaps focus also your spectrum of antibiotics much better this way. Or the other way to do is to actually go in direction of parenteral release. I mean, you have also some sterile filtered products. You can make sterile filtration of the product, and then you might use a filter integrity test that might be used as your parametric release parameter.
Thank you, David. The next question is, will it work when the product is live human cells like CAR T cells?
Yeah, so there are already some methods which are widely used, which are based on CO2 detection. It's the growth-based method. So this is now widely used for cell cultures, including the CAR T cells, as a release testing of a sterility test. Generally, the people who are applying this method have a release time of 5 to 7 days.
Thank you so much, David. Before we finish the webinar, I would like to ask our presenters, David and Erin, if they have any closing remarks. David?
Yes, so thank you for giving me the final word on my presentation. Well, I just want to say that the actual compendial sterility test has first been written in the USP since 1936. So now we are at the time of SpaceX and flying taxis in the next years or so, and we're still remaining with this very old method. So we also have in parallel new products, new therapies, which are very potent, which are really profitable for the patient, and it's really important that the people who are writing the standards and also the legislator accompanies this movement. So now we have started this. I'm really happy that the Microbiology Expert Committee has started this journey. It's going to be complex because we don't have systems which can really be applicable as such. We need to make further analysis, but the journey has started, and I hope that we're going to have in the next years or so a compendial chapter which can really support the sterility testing of all these new advanced therapeutical medicine.
Thank you, David. Erin, do you have any closing remarks?
Yes, I mean, I couldn't echo that enough. I think it is so wonderful to see that those who are writing the standards, in this case the USP, has turned to an expert committee to move towards, taking a closer look at what can be done to maybe a little bit lower the bar on what it would take for a company to adopt a rapid sterility test, since there's so much advantage to be gained. It's really a great move towards encouraging the adoption of rapid sterility testing to see these USP activities. And so we look forward to hearing more updates and to following the progress of this committee, and I wanted to also thank all the attendees today, and also to Biopharma Asia for hosting the webinar. It's been a pleasure to participate, and so thank you very much.
Thank you. I would like to take this opportunity to thank our presenters, Dr. David Roesti and Erin Patton, for sharing their knowledge with us. I would like to remind our audience that they can view this webinar on demand by visiting biopharma-asia.com. Thank you, everyone.